Okay, since nobody has joined the session, we'll start the session, and this will be recorded for people to see it later. So I'll get started now. So similar to the previous live sessions, the format remains the same. I'll discuss MCQs, and then I'll explain, go through each of the options, and I'll explain why a certain option is correct or wrong. So last week we had stopped. We were doing week six MCQ questions related to week six videos, and we had done nine. We had solved nine MCQs, and we had solved. We had stopped at this particular uh, place. So we'll continue from here. Select the correct sequence. Uh, so this is, I think, related to pre-programmed reactions. So what happens? What is the correct sequence in which certain events occur? So A, T reflex, pre-programmed reaction, voluntary action. B, pre-programmed reaction, voluntary action, T reflex. C, T reflex, voluntary reaction, pre-programmed reaction. Or D, voluntary reaction, T reflex, pre-programmed reaction. So let me just. So we have seen that in normal everyday movements, let us say a motor command is given from the higher centers, but after the motor command has been given, let us say that there is some change in the environmental conditions. So now that motor command is not relevant anymore. So there can be some errors in the movement, and then these errors need to be corrected quickly, and that is where these pre-programmed reactions come into the picture. For example, we face this every day. For example, if you are walking on the road or on the footpath, uh, we are giving the brain the appropriate commands. The brain is giving the body the appropriate commands to walk, but there can be perturbations, various kind of perturbations. So it can be, for example, a bad if the footpath is bad, then there might be some holes or some uneven path, or for example, somebody comes in between while we are walking. So then we have to make we have to make corrective movements to avoid falling down or bumping into somebody. And that is where these pre-programmed reactions come into play. So they take corrective action. So to a pre-programmed reaction does not need a particular any one stimulus to trigger it. The modality of the stimulus does not matter. It can be a visual stimulus, it can be a haptic stimulus, it can be a light stimulus. Uh, so the stimulus can be anything to trigger the pre-programmed reaction, but the reaction remains the same. That is a brief explanation of pre-programmed reaction. So let us consider a situation uh, where a person is holding a particular object against weight. Uh, you, so one so the the instruction to the participant is to maintain the position of the joint, is to maintain the position of the joint in case there is a perturbation, and the person is holding a particular weight. So now, if a perturbation is given, uh, there is some corrective reaction that happens. So let us go to the. I'll just show you what I mean. So this is the situation that happens. Uh, so an unexpected perturbation of a joint gives rise to a sequence of EMG events in a muscle. So we can see that perturbation, by perturbation I mean there's some unexpected movement that happens. So the task is to maintain uh, the hand in a particular position, and the person is holding uh, a particular object. Uh, so when this perturbation is given, initially there's a monosynaptic reflex. So this M1 response. Uh, so the latency is of this particular response, which is seen in the EMG, is similar to that of a monosynaptic reflex, and that is followed by a pre-programmed reaction. So M2, M3. So this is the this this particular reaction is a corrective measure to so the instruction was to maintain the object, maintain the hand in the initial position. So this tries to is a corrective reaction when the perturbation is given, so that that instruction is followed. And then it since the pre-programmed reaction is always the same, uh, there still needs to be some voluntary movement to finish the corrective reaction. So the sequence is monosynaptic is followed by a pre-programmed reaction followed by a voluntary reaction. So let us go back. So a T reflex is a monosynaptic reflex. So we just saw T reflex followed by, so this is the voluntary reaction, and this is M2, M3. This is the pre-programmed reaction, PPR, and this is monosynaptic reflex, and this is the perturbation, and this is time. So the answer is A. T reflex, that is T reflex is a monosynaptic reflex followed by a pre-programmed reaction followed by a voluntary action. Which of the following is true with regard to response time? So again, you can pause the video. Uh, you can pause the video and then try and answer this question. Uh, so we have seen that. So usually a T reflex the is a monosynaptic reflex, and it takes around 20 to 30 milliseconds. So we can eliminate this option. And M reflex is even smaller than the latency is even lesser than T reflex. So again, this is not correct. And we know that a pre-programmed reaction, the latency of a pre-programmed reaction is somewhere in between a voluntary reaction and an M2, M3. Has somebody joined in? Stay in call. So a pre-programmed reaction, we know that for a pre-programmed reaction, which is here in this case M2, M3, its latency is somewhere in between a monosynaptic response and a voluntary response. So the answer is C. A voluntary reaction takes slightly more time. So this is not the answer. Answer is C. Okay. And we can see that in this image here as well. So you can see the timeline around 60 to 70 milliseconds. Around that time we start to see the pre-programmed reaction. Which of the following is true? Mesencephalic preparation is a type of decerebrate preparation where spontaneous motor activity is seen. Okay. Uh, so mesencephalic preparation is a type of decerebrate preparation, that is correct. Where spontaneous motor activity is seen, this is not correct. So in this kind of preparation, an electrical stimulation is required to see any motor activity. Uh, a pre-mammalian preparation is a type of decerebrate preparation, that is correct, where electrical stimulation of midbrain and locomotor region is required. Again, this part is wrong because uh, in this kind of preparation, spontaneous motor activity is seen. So we can eliminate these two. So these are actually opposite the explanation of these two. In deafferentated preparation, dorsal root ganglion is dissected. Yes, we want to remove any sensation any sensation that is going to the brain. Uh, so any touch sensation or haptic sensation or any other. We don't want afferent inputs to go to the brain, and hence and we know that these afferent inputs go into the brain through the dorsal root of the spinal cord, whereas the efferent inputs or the motor commands go out from the spinal cord through the ventral root ganglion. So this is correct. In spinal preparation, drugs like levodopa, L-dopa and nilamide is used to create locomotor-like movements. This is also correct. We have seen this. So let us look at if we have the slides. So spinal cord is removed from a neonatal rat. In spinal preparation, the spinal cord is transected at the lower thoracic level, isolating the spinal segments that control the hind limb from the rest of the central nervous system. So there are two types of spinal preparations. In the acute type of spinal preparation, these drugs are used. So the answer is C and D. Okay, studies of Ed Evarts showed that A. Neuron encodes movement kinetics when kinematics was constant. The neuron encodes movement kinematics when kinetics was held constant. Some neuron in the wrist area in the motor cortex, which was active during flexion, was silent during extension. Some neuron in the wrist area in the motor cortex, which was active during flexion, was also active during extension. So let us go back and see what Ed Evarts, the actual experiment that Ed Evarts had done. So this will refresh the memory. Let us discuss this. So, so this is the experiment that Ed Evarts had done. So he took up. We know that Herbert Jasper developed the microelectrode stimulation technique, where a single electrodes, microde electrode, single single neuron electrode recordings could be taken as well as stimulation could be given. So this was the contribution of Herbert Jasper, and this technique was used by many scientists to carry out various experiments. So what Ed Evarts did was uh, in Ed Evarts studies, he took recording from a neuron in the wrist area of the motor cortex for various conditions. So what he observed was during flexion. So this is the wrist position. Yeah. So this is extension and this is flexion. Uh, So when the neurons were flexing and extending, uh, we can see here that, see, so the explanation is neurons, the monkey performed flexion of the wrist, and the electrode recordings were taken, and the neurons, it was found that neurons were active during flexion, were reciprocally silent during extension, and vice versa. So, for example, let us say that uh, this is the wrist position, which is at the extended position. Uh, so here the monkey starts flexing. However, during extension, the same neurons, that same neuron is not active. So this was what Ed Evarts had done, and also he did one more experiment. So let us look at that as well. So here, what he did was, uh, so these are the EMG activities. The monkey holds a particular bar, and all the other regions of the body are restrained for the monkey. And then recordings from the neurons in the wrist area of the primary motor cortex is taken. So we can see here that from the extended position, the monkey flexes. The monkey does the flexion action, and we can see that during extension, uh, uh, this is what the neuronal activity looks like. Okay, and now a particular weight is added here. So this opposes the flexion action, right? So we can see here that the extensor muscles are completely silent, whereas the flexor muscles become very active. And we can see that before, see over here as well, before the flexion actually takes place, we can see that there is neuronal activity. So these are the commands that are going to this particular muscle. Similarly, here we can see that there's a burst of activity, the neuronal activity, before the flexion starts. So this is the command that is going, and we can see that the frequency of the firing is much higher in this case. This is because there is a weight that is opposing the flexion. And similarly, in this particular case, what happens is there is a weight that is assisting the flexion. So there is no requirement as such. Uh, so there is no requirement as such for the flexor muscles to actually contract, because the weight is assisting that contraction. Uh, However, we need extensor muscles so that to be active, so that we don't flex beyond a particular uh, flex beyond a particular range. So that is why flex. We can see that there's some flexor activity. So initially we need to hold the flexion, we need to prevent the flexion, and then when the instruction is there, we need to flex. The monkey flexes. So we can see here that the wrist displacement is the same, but the neuronal activity. So in all the three cases, the wrist displacement is the same, but the neuronal activity changes as the loads and compensatory muscle activity change. So thus the activity of the motor cortex neuron is better related to the direction and level of forces, and the muscle activity exerted during the movement than the direction of the wrist displacement. So here we are actually coding, what is being coded is the kinetics or the forces, or that the muscle generates, not the displacement. So we can see in all the three cases that the displacement is the same, but due to the various conditions, the forces that need to be generated by the flexor muscles uh, changes. So the correspondingly the neuronal command that is going to these muscles also is changing, as we can see in these three plots here. So this is the situation. So now let us look. discuss this particular question. The neuron encodes movement kinetics when kinematics was constant. This is correct because kinetics is the forces, whereas the kinematics in this particular example, the kinematics is the displacement. So the displacement was constant in all the three cases, but the movement kinetics or the forces that needed to be generated had to be different uh, in all the three cases. So this is correct. The neuron encodes movement kinetics when kinematics when kinetics was held constant. This is not correct. Uh, it's the opposite. Some neuron in the wrist area in the motor cortex, which was active during flexion, was silent during extension. We also saw this. This is what Ed Awards had shown in his very first experiment. And some neuron in the wrist area in the motor cortex was active during flexion, was also active during extension. This is false. So the answer is A and C. Okay. So now, which of the following is false? I'll just step out for a minute uh, and come back. I need to get some water. Till then, you can try and solve this question. Okay, let's discuss this question now. Uh, Pre-supplementary area and pre-dorsal which of these are false? So pre-supplementary area and pre-dorsal premotor area receive very weak inputs from the somatosensory cortex, but strong inputs from the prefrontal cortex. If you recall the explanation from the class, we know that this statement is correct from the recorded videos. Then you know that the statement is correct. Then the dorsal premotor cortex and ventral premotor cortex communicates with parietal cortex and provide context specificity. This part is correct. They do communicate with the parietal cortex, but they don't provide uh, they don't provide context specificity. But rather, I think they provide movement. They are involved in movement adjustment. So this part is wrong. So this is a false statement. Pre-supplementary motor area and pre-dorsal pre-motor area communicate with the prefrontal cortex. That is correct, and it is involved in movement adjustment. This is false. So they're not involved in movement adjustment, but they provide context specificity. So I think these two have been interchanged. So this is a false answer. Primary motor cortex and the supplementary motor area communicate with primary somatosensory cortex and is involved in movement mechanics. Yes, this is correct. They do communicate with the this thing and the somatosensory cortex, and they are directly involved in movement mechanics. So the question is, which of the following statements are false? So B and C are false. So the answer is B and C. Okay, so this is a big statement, a big question. Select the correct statement. Basal ganglia is involved in movement selection and communicates directly with the spinal cord. Okay, let us just open the first slide. So this is what these are the neural centers responsible for movement. Uh, so this is what happens. This is a brief bird's eye view of what is happening. Of course, there's much more complexity. So the basal ganglia is involved in movement selection, that is correct, but they communicate with the motor cortex and the brain stem. So let us see what this option was. So we have to select the correct statements, right? Uh, so they are involved in movement selection, but they do not communicate directly. But they do not communicate directly with the spinal cord. So this part is wrong. It's saying they communicate, but that is not the case. So cerebellum is involved in error correction and receives information along the spinocerebellar pathway. This is correct. So we can go ahead and see that slide. So they directly communicate. Communicate with the motor cortex. The cerebellum is involved in error correction. They communicate with the motor cortex, and they also communicate with directly with the spinal cord through the spinocerebellar pathway. Basal ganglia is involved in movement selection and communicates to the spinal cord via the primary motor cortex. That is correct. We have seen that image. Let me just open it again. We can see the basal ganglia communicates with the motor cortex, and the motor cortex in turn communicates with the spinal cord. So uh, this is the spinal cord actually. This entire area. Pre-supplementary motor area and pre-dorsal premotor area do not have direct connections to the primary motor cortex, but they have connections to the prefrontal cortex. Uh, yes, this is true. So the pre-supplementary motor area, which is more rostral to the supplementary motor area proper, and the pre-dorsal premotor cortex, which is again more rostral to the dorsal premotor cortex. So they don't have direct connections to the primary motor cortex, but they have reciprocal connections to the prefrontal cortex. That is correct. So again, this answer is correct. Cerebellum receives information only along the spinocerebellar pathway and does not communicate with the primary motor cortex. This is false. We have just seen it that it communicates with the uh, it communicates both with the primary motor cortex as well as the spinal cord. The primary motor cortex transmits information to the alpha motor neuronal pool of the skeletal muscle via the uh, local neuronal circuit. This is correct. So let us again, we have the local neuronal circuit, but we also have some uh, uh, neurons from the motor cortex that directly synapse onto the alpha motor neurons of uh, uh, of the muscles which supply the muscle. So these are they have direct monosynaptic connections, and uh, uh, so these mono so they can we have the story is that there are many connections to these neurons, the neurons from the motor cortex synapse with many interneurons, and then these interneurons in turn there is there are various levels of hierarchy, and then they finally give the signal to the alpha motor neurons. But these there are some neurons, especially the ones controlling the hand and finger muscles, which synapse directly to the alpha motor neurons. So these are the monosynaptic projections, and this is seen more and more in humans when compared to lower mammals. So lower mammals have less uh, monosynaptic connections. Sorry, has somebody joined in? Madhav. Okay, Madhav has joined in. So Madhav, hello, welcome to the live session. So I think you are the only participant who has joined in.
Live session. So I think you are the only participant who has joined. Oh, okay, sir.
Yeah. So you can just mute yourself. So what I was doing was usually in the live sessions, I don't know whether you have attended previous live sessions. So what I do is I have a set of MCQs that I discuss, and then I give you a few minutes to attempt the MCQ, and then I'll go through each of the options and discuss the concepts related to it. So right now, since nobody else had joined in, I had so since this videos will be recorded, I had continued to take the live session so that people can see the recorded video later. So you can attempt to answer the question. So I'm currently in question 15. So you can I'll give you like a couple of minutes to answer this. I'll just remove these and then we'll discuss this.
Sir, I'm not able to see the screen, sir.
Oh, you're not able to see the
Oh, you're
screen,
not
is it?
able to see the screen, is it? Yes, sir.
Okay, 1 second. Yeah, I hope you can see the screen now.
Yes, sir. Yes, sir.
Yeah, so I was discussing this particular question. So you can take a couple of minutes. So I discussed a few questions from here. So I won't go through these questions again, but you can see the explanation in the recorded video. Uh, so for now, you can try and attempt to answer this particular question, and then we'll discuss this. So you can type your answer in the chat box. I'll set an alarm for 2 minutes, following which we'll discuss this question. ABG. Okay. ABG. Okay. So let us discuss this. So select the correct statement. Basal ganglia is involved in movement selection and communicates directly with the spinal cord. So I'll just open up this slide. So this is a bird's eye view of what is happening in the motor movement control system. So we have the motor cortex, the basal ganglia, cerebellum, and the spinal at the level of the spinal cord, what is happening, and then finally the motor output to the skeletal muscles. So we can see that here that basal ganglia is involved in movement selection. That part is correct. However, it communicates to the motor cortex. It doesn't communicate directly with the spinal cord. So this particular thing is wrong. Uh, Cerebellum is involved in error correction and receives information along the spinocerebellar pathway. So if we go back to the slide, we can see that cerebellum is indeed involved in error correction, and it communicates with the spinal cord through what is called as the spinocerebellar pathway. So this option is correct. Basal ganglia is involved in movement selection and communicates to the spinal cord via the primary motor cortex. This is correct. So if we look at this, we can see that the basal ganglia is communicating with the motor cortex, and it is involved in movement selection. Uh, Pre-supplementary motor area and pre-dorsal premotor area do not have direct connections to the primary motor cortex, but have reciprocal connections to the prefrontal cortex. So in the lecture, sir has mentioned this, that they don't have direct connections to the primary motor cortex, but they do have reciprocal connections with the prefrontal cortex. Uh, so this is also correct, this particular. So pre-supplementary motor area is the area that is rostral to the supplementary motor area proper, and pre-dorsal premotor area is the area rostral to the dorsal premotor area. Uh, okay. Cerebellum receives information only along the spinocerebellar pathway and does not communicate with the primary motor cortex. This is a false statement. So we can see that here. Uh, So here we can see that the cerebellum is communicating both with directly with the spinal cord through the spinocerebellar pathway as well as the motor cortex. So this is false. Uh, the primary motor cortex transmits information to the alpha motor neuronal pool of the skeletal muscle via the local neuronal circuit. Yes, this is correct. However, we should also remember that there are certain neurons called the upper motor neurons or the corticospinal neurons, which directly synapse with the alpha motor neurons, supplying especially the hand muscles, hand and finger muscles. So that is a speciality which is seen especially in humans and not in lower mammals. So in humans, we see that the area of the brain controlling the hand muscles make monosynaptic connections to the alpha motor neuron, uh, which supply these hand and finger muscles. So there is also these neurons which synapse directly with the alpha motor neuron. Uh, and if we look at lower mammals, in the lower mammals, these monosynaptic connections are very few. But if we start going higher, especially in the macaque, the number of these upper motor neurons or the corticospinal motor the corticomotor neurons is more, and it is the highest in humans. So that is why studying hand and finger function can give us insights into the control strategies employed by the central nervous system in controlling movements when compared to other areas. For example, if we are doing research, uh, some research related to control of legs or other areas in the body, then there is more level of interference. So the commands are sent to the spinal cord, and then there is some processing happening in the spinal cord, and then the spinal cord, after various levels of hierarchy, then sends the motor commands to the alpha motor neurons of the other parts of the body. But in the case of hands, there is direct monosynaptic connection from the motor cortex to the alpha motor neurons supplying the hand muscles. So, in that sense, it is advantageous to study hand and finger movements. Uh, and So, let us go back to the question. The alpha motor, why the so this is correct. Pre-supplementary motor area and pre-dorsal premotor area have direct connections to the primary motor cortex, but do not have reciprocal connections to the prefrontal cortex. Uh, no, this is not true. We have seen that they don't have direct connections to the primary motor cortex, right? Similarly, here also, so this is also similar. Pre supplementary motor area and pre dorsal premotor area uh, have direct connections to the primary motor cortex. Uh, Neither have direct connections to the primary motor nor have reciprocal connections to the prefrontal cortex. This part is wrong. So the answer should be B, C, D, and F. Let me erase this. Okay. Uh, Going to the next question. Which of the following statements is true based on historical studies? So you can again take a couple of minutes to answer this, and then we'll discuss this question. Okay, let's see what the response is. Okay, Madhav was saying C. Let's see. So Madhav's response is C. Okay, so let us recall these terms. So the rostral in the brain. So let us say this is the eye here. The rostral region is this in this direction, and caudal region is here. Uh, so we know that the area immediately rostral to the central sulcus is the primary motor cortex. So this is correct. Uh, so we have the central sulcus, and immediately rostral to it we have the primary motor cortex, and immediately caudal to it we have the somatosensory cortex. Uh, region caudal to the central sulcus is primary motor cortex. That is false. The primary motor cortex has a distinct structure and lacks the granule cells other parts of the cortex has. Yes, this is correct. That is why the primary motor cortex is the agranular cortex. I think I forgot who was the one who discussed. I think it was Campbell, if I'm not wrong. Okay, so we'll go to the next one. I think it was Broadman and Campbell who first identified this. Uh, then D, the primary motor cortex has a distinct structure and has granule cells that other parts of the cortex do not have. This is false. Uh, Region that is caudal to the supplementary motor area is the primary motor cortex. Yes, this is correct. So immediately rostral to the to the primary motor cortex, we have the supplementary motor area. So if we look at it from the point of view of the supplementary motor area, then the area immediately caudal, that is towards the posterior end, is the primary motor cortex. Uh, the region that is caudal to the supplementary motor area is the premotor cortex. So the premotor cortex is actually the supplementary motor area is a part of the premotor cortex. So this is false. And the region of the brain that causes problems with speech production and speech perception when affected was called as Broca's area and Wernicke's area. Uh, yes, this is correct because we know that Broca and Wernicke were the first one to localize function in the brain. So, a particular region in the brain is associated with the particular function. So this is correct. This was the first. I think this was the first case where they localized. They said that a particular area in the brain is responsible for a particular function, and in that time, this idea was revolutionary because uh, the traditional view was that the entire brain is involved in doing everything. Uh, so they challenged that notion with their claims. And Ed Evans performed experiments on live behaving dogs. That is not true. Uh, I think it was Clinton Woolsey who performed the experiments on living dogs, not Ed Edwards. Ed Edwards had done experiments on monkeys. So the answer is C. Uh, A, C, E, and G. So this is the answer. Okay. So we are done with this week's questions. We have some more questions. Okay, we'll discuss one last question and then we'll continue next week. So question 2, as per Graziano's experiments, so you can take around 30 seconds to answer this and then we'll discuss this. Okay, let's see what the response is. Okay, Madhav is saying B. Okay, so let us recall what Graziano did. So Graziano, what he did was usually the studies involving the brain were stimulation-based studies where very small duration stimulations were given or recordings were taken. But what Graziano did was something completely different and which was against the traditional neurophysiological approach. What he did was he gave long in the motor cortex, in regions of the motor cortex, he gave stimulation for very long periods of time in monkeys. uh, Stimulation in the for the order of 500 milliseconds. So usually what the stimulation duration is 1 millisecond, 2 milliseconds, etc. But what Graziano did was, I mean, it's controversial as well as something completely different to the traditional neurophysiological approach. And after giving such a long duration of 500 millisecond stimulation, what he observed uh, well coordinated movements in the monkey. For example, the monkey made certain movements from the hand to the mouth, or made certain defensive stances. So the answer is well-defined movements like hand-to-mouth trajectories are observed during stimulation of neurons for a long duration. Okay, so answer is C. The previous studies, various studies by Georgopoulos, Ed Evarts, Eb Fetz, and Kalaska, and all these investigated what exactly is the motor cortex encoding. Is it encoding kinetics of the movement or the kinematics of the movement? But in this particular case, so they showed through their experiments that the neurons are coding direction, kinetics, kinematics, etc. But Graziano's work shows that uh, adds another dimension to this. It shows that the motor cortex, the neurons are actually coding movements, entire movements itself. So it's unclear exactly what the motor cortex is coding. Is it kinetics, kinematics, or entire movements itself? So that's still a question mark, and it's still being researched. Uh, so we'll stop the session here,
The session here.
and
And
if you have any question, you can ask me now, Madhav.
if you have any question, you can ask me now, Madhav.
No, sir, no, sir.
Okay, okay, Madhav. Thank you for joining in, and see you next week.